prominent Yale professor explains how Darwin's theory of evolution does not match the science. Alexander Light of Humans Are Free reports, science never ceases to question. When a theory is taught as an unquestionable fact, it should be quite obvious that something is wrong. Today's science isn't really science, and this is not only true for topics such as evolution, it's true in many areas where science is used for an agenda by powerful and corrupt forces. Health sciences are a great example. As Bud Relman, former editor of New England Journal of Medicine, said, quote, "The medical profession is being bought by the pharmaceutical industry, not only in terms of the practice of medicine, but also in terms of teaching and research. The academic institutions of this country are allowing themselves to be the paid agents of pharmaceutical industry. I think it's disgraceful," end quote. Today, some scientific publications are silenced and others are pushed forward, depending on how they affect corporate and political agendas. It's not actually about the science. What the mainstream media preaches are settled science is not actually settled. In fact, it's often highly dubious. Why don't more people see this? The answer is simple. It's because we rely on outside sources to tell us what is, instead of taking the time as individual researchers to really look into something. The theory of evolution falls into this category, scientists who have rejected the basic premises of Darwin's theory continue to be condemned and shunned by the mainstream community and powerful people. This is because their paradigm-shifting thoughts and ideas on the subject, though more grounded in fact, threaten the goal of the global elite, which NSA whistleblower William Binney says is total population control. It's because their paradigm-shifting thoughts and ideas on the subject, though more grounded in fact, threaten the goal of the global elite. The average person who gets a bachelor's degree in science is trained to simply repeat the same old textbook rhetoric as to why evolution is to be the be-all and end-all of human existence, without actually looking into why the theory is highly questionable. One of the latest dissidents is David Gelernter, prominent scientist and distinguished professor of computer science at Yale University. He recently published an essay in the Claremont Review of Books explaining his objections to a premise behind Darwin's theory. He first points to the famous Cambrian explosion, quote unquote, which occurred 500 million years ago, in which a number of new organisms, including the first ever known animals, pop up suddenly in the fossil record over a period of approximately 70 million years ago. Apparently, this giant explosion of spontaneous life was followed by evolution, slow growth, and scanty fossils, mainly of single-celled organisms dating back to the origins of life roughly 3.5 billion years ago. From here, he explains how Darwin's theory predicts that new life forms evolve gradually from preceding ones, but if this is applied to the Cambrian creatures as well, it does not work. The predecessors to the Cambrian creatures are missing, something that Darwin himself was disturbed by as well. Furthermore, even without this fact, many scientists have already used other aspects of the fossil record to demonstrate that Darwin's theory is clearly wrong. Quote, "The Cambrian explosion had been unearthed, and beneath those Cambrian creatures, their Precambrian predecessors should have been waiting, and were not. In fact, the fossil record as a whole lacked the upward branching structure Darwin predicted. The ever-expanding fossil archives don't look good for Darwin, who made clear and concrete predictions that have so far been falsified, according to many reputable paleontologists anyway." Quote, "When does the clock run out on those predictions? Never. But any thoughtful person must ask himself whether scientists today are looking for evidence that bears on Darwin or looking to explain away evidence that contradicts him. There are some of each. Scientists are only human, and their thinking, like everyone else's, is colored by emotion. The genesis of new life forms." His next point goes a little deeper. Many people point to the fact that variations occurs naturally among individuals and different traits are passed on. This is something observable and something that we all know. Many scientists actually use this point as a proof for evolution, which does not make much sense. According to proponents of the theory of evolution, natural variation is a consequence of random change or mutation to cells, to the genetic information within our cells that deal with reproduction. These cells pass on genetic change to the next generation, which according to Darwinians changes the future of the species and not just the individuals. The engine behind this thought, as Gellner explains, is change, driven by the survival of the fittest and obviously lots and lots of time. He then goes on to ask a very crucial question. What exactly does generating new forms of life entail? Many within the field agree that generating a new shape of protein is the key to it, but does Darwinian evolution even purport to be able to do this? For Chris Williams, a PhD biochemist at Ohio State University, the full scope of Darwinian evolution barely touches upon this important matter. Quote, as a biochemist and software developer who works in genetic and metabolic screening, I am continually amazed by the incredible complexity of life. For example, each of us has a vast computer program of 6 billion DNA bases, and every cell that guided our development from a fertilized egg specifies how to make more than 200 tissue types and ties all this together in numerous highly functional organ systems. Few people outside of genetics or biochemistry realize that evolutionists still can provide no substantial details at all about the origin of life, and particularly the origin of genetic information in the first self-replicating organism. What genes did it require? Or did it even have genes? How much DNA and RNA did it have? Or did it even have nucleic acids? How did huge information-rich molecules arise before natural selection? Exactly how did the genetic code linking nucleic acids to amino acid sequence originate? Clearly, the origin of life, the foundation of evolution, is still virtually all speculation and little, if no fact. End quote. Intelligent design. More and more, the evidence points to the great intelligence apparent in the system of life creation. The reason that Darwinian evolution is being left behind, and for many is obsolete, is because it is completely based on random, non-intelligent processes. Edward Peltzer, PhD Oceanography, University of California, San Diego, Scripps Institute, Associate Editor, Marine Chemistry, uses a clear, real-life laboratory example to explain the need to posit the existence of an overriding, quote, intelligence, end quote, in order for things to make any sense. He says, as a chemist, the most fascinating issue for me revolves around the origin of life. Before life began, there was no biology, only chemistry. And chemistry is the same for all time. What works or not today worked or not back in the beginning. So our ideas about what happened on Earth prior to the emergence of life are eminently testable in the lab. And what we have seen thus far, when the reactions are left unguided as they would be in the natural world, is not much. Indeed, the decomposition reactions and competing reactions outdistance the synthetic reactions by far. It's only when an intelligent agent, such as the scientist or graduate student, intervenes and tweaks the reaction's conditions just right, do we see any progress at all. And even that is still quite limited and very far from what we need to get to. Thus, it is the very chemistry that speaks of a need for something more than just time and chance. And whether that be simply a highly specified set of initial conditions, fine-tuning, or some form of continual guidance until life ultimately emerges is still unknown. But what we do know is the random chemical reactions are both woefully insufficient and are often working against the pathways needed to succeed. And for these reasons, I have serious doubts about whether the current Darwinian paradigm will ever make additional progress in this area. Gellner brings this conversation specifically to the generation of proteins. Quote, proteins are the special ops forces, or maybe the Marines, of living cells, especially that they are common instead of rare. They do all the heavy lifting, all the tricky and critical assignments in a dazzling range of roles. Proteins called enzymes, catalysts, all sorts of reactions and drive cellular metabolism. Other proteins, such as collagen, give cells shape and structure like tent poles, but in far more shapes. Nerve function, muscle function, and photosynthesis are all driven by proteins. And in doing these jobs and many others, the actual 3D shape of the protein molecule is important. So, is the simple neo-Darwinian mechanism up to this task? Are random mutations plus natural selection sufficient to create new protein shapes? Diving into the proteins. Gelantner goes on to answer that question in great detail, and after going through the entire explanation, he comes to what seems to be an inarguable conclusion: that the theory of evolution cannot, in any way, be a possible explanation for the generation of new proteins and mutations that are required for evolution to occur at all. This explanation is complex, but well worth it if you really want to understand how the theory of evolution is refuted by the science of proteins. Quote, how to make proteins is our first question. Proteins are chains, linear sequences of atom groups, each bonding to the next. A protein molecule is based on a chain of amino acids. 150 elements is a modest-sized chain. The average is 250. Each link is chosen ordinarily from one of 20 amino acids. A chain of amino acids is a polypeptide, peptide being the type of chemical bond that joins one amino acid to the next. But this chain is only the starting point. Chemical forces among the links make parts of the chain twist themselves into helices, others straighten out, and then sometimes jackknife repeatedly, like a carpenter's rule, into flat sheets. Then the whole assemblage folds itself up like a complete sheet of origami paper. And the actual 3D shape of the resulting molecule is, as I've said, important. Imagine a 150 element protein as a chain of 150 beads, each bead chosen from 20 varieties. But only certain chains will work. Only certain beads combinations will form themselves into stable, useful, well-shaped proteins. So how hard is it to build a useful, well-shaped protein? Can you throw a bunch of amino acids together and assume that you'll get something good? Or must you choose each element of the chain with painstaking care? It happens to be very hard to choose the right beads. Inventing a new protein means inventing a new gene. Enter, finally, genes, DNA, etc. with suitable fanfare. Genes spell out the links of a protein chain, amino acid by amino acid. Each gene is a segment of DNA, the world's most admired micromolecule. DNA, of course, is the famous double helix or spiral staircase, where each step is a pair of nucleotides. And as you read the nucleotides along one edge of the staircase, sitting on one step and bumping your way downwards to the next and the next, each group of 3 nucleotides along the way specifies an amino acid. Each 3 nucleotide group is a codon, and the correspondence between codons and amino acids is a genetic code. The 4 nucleotides in DNA are abbreviated T, A, C, and G, and you can look up the code in a high school textbook. TTA and TTC stand for phenylalanine, and TCT for serine, and so on. Your task is to invent a new gene by mutation. By the accidental change of one codon to a different codon, you have 2 possible starting points for this attempt. You can mutate an existing gene or mutate gibberish. You have a choice because DNA actually consists of valid genes separated by long sequences of nonsense. Most biologists think that the nonsense sequences are the main source of new genes. If you tinker with a valid gene, you will almost certainly make it worse, to the point where its protein misfires and endangers or kills its organism long before you start making it better. The gibberish sequence, on the other hand, sits on the sidelines without making proteins, and you can mutate them, so far as we know, without endangering anything. The mutated sequence can then be passed on to the next generation, where it can be mutated again. Thus, mutations can accumulate on the sidelines without affecting the organism. But if you mutate your way to an actual valid new gene, your new gene can create a new protein and thereby potentially play a role in evolution. Mutations themselves enter the picture when DNA splits in half down the center of the staircase, thereby allowing the enclosing cell to split in half and the encompassing organism to grow. Each half staircase summons a matching set of nucleotides from the surrounding chemical soup. Two complete new DNA molecules emerge. A mistake in this elegant replication process, the wrong nucleotide answering the call, a nucleotide typo, yields a mutation either to a valid blueprint or a stretch of gibberish. Building a better protein? Now, at last, we're ready to take Darwin out for the test drive. Starting with 150 links of gibberish, what are the chances that we can mutate our way to a useful new shape of protein? We can ask basically the same question in a more manageable way. What are the chances that a random 150 link sequence will create such a protein? Nonsense sequences are essentially random. Mutations are random. Make random changes to a random sequence and you get another random sequence. So close your eyes, make 150 random choices from your 20 bead boxes and string up your beads in the order in which you choose them. What are the odds that you come up with a useful new protein? It's easy to see that the total number of possible sequences is immense. It's easy to believe, although non-chemists must take their colleagues' word for it, that the subset of useful sequences that create real usable proteins, is in comparison tiny. But we must know how immense and how tiny. The total count of possible 150 link chains, where each link is chosen separately for 20 amino acids, is 20,150. In other words, 20,150 roughly equals 10,195, and there are only 1,080 atoms in the universe. What proportion of these many polypeptides are useful proteins? Douglas Axe did a series of experiments to estimate how many 150 long chains are capable of stable folds, of reaching the final step in the protein creative process, the folding and of holding their shapes long enough to be useful. Axe is a distinguished biologist with 5-star breeding. He was a graduate student at Caltech, then joined the Center for Protein Engineering at Cambridge. The biologists whose work Meyer discusses are mainly first-rate establishment scientists. Quote, he estimated that of all 150 link amino acid sequences, 1 in 1074 will be capable of folding into a stable protein. To say that your chances are 1 in 1074 is no different, in practice, from saying that they are 0. It's not surprising that your chances of hitting a stable protein that performs some useful function and might therefore play a part in evolution are even smaller. Axe puts them at 1 in 1077. In other words, immense is so big and tiny is so small that neo-Darwinian evolution is so far a dead loss. Try to mutate your way from 150 links of gibberish to a working, useful protein, and you are guaranteed to fail. Try it with 10 mutations, 1,000, a million, you fail. The odds bury you, and it can't be done. Proteins, mutations are one of several issues. Despite all of the scientific dogma that plagues this issue, proteins and mutations and lack of fossil evidence are simply the tip of the iceberg when it comes to finding faults found within the theory of evolution. There are many facts, information, science, and new discoveries that would make one wonder how it's even still being taught. Furthermore, despite the fact that we get pounded with the idea that random mutation is ultimate truth within the mainstream and that one is wrong for questioning it, there are a number of prominent scientists who are actually getting together in large numbers to collectively refute Darwinism. A group of 500 scientists from several fields came together a few years to create a scientific dissident from Darwinism, quote unquote. As one example, the issue is that these scientists are never getting any mainstream attention, but clearly there are some very intelligent people here. Quote, the theory will be with us for a long time, exerting enormous cultural force. Darwin is no Newton. Newton's physics survived Einstein and will always survive because it explains the cases that dominate all of space-time except for the extreme ends of the spectrum at the very smallest and largest scales. It's just these most important cases, the ones we see all around us, that Darwin can't explain. Yet his theory does explain cases of real significance, and Darwin's intellectual daring will always be inspiring. The man will always be admired. He now poses a final challenge. Whether biology will rise to this last one, as well as it did to the first, when his theory upset every apple cart, remains to be seen. How cleanly and quickly can the field get over Darwin and move on, with due allowance for every Darwinist having to study all the evidence for himself? There is one of the most important questions facing science in the 21st century. Other examples that throw off the theory of evolution. Not long ago, I wrote about a recent paper published by 33 scientists in the Progress in Biophysics and Molecular Biology journal, suggesting that the flourishing of life during the Cambrian era, the Cambrian explosion, originates from the stars is so fascinating. With the rapidly increasing number of exoplanets that have been discovered in the habitable zones of long-lived red dwarf stars, according to Golet and colleagues in 2016, the prospects for genetic exchanges between life-bearing Earth-like planets cannot be ignored. There is a great little blurb from Cosmos magazine, one of the few outlets who are talking about the study. With 33 authors from a wide range of reputable universities and research institutes, the paper makes a seemingly incredible claim. The claim that, if true, would have the most profound consequences for our understanding of the universe. Life, the paper argues, did not originate on the planet Earth. The response? Near silence. The reasons for this are fascinating, as the evidence and claims advanced by the paper itself, entitled Cause of the Cambrian Explosion, Terrestrial or Cosmic. The publication revives a controversial idea concerning the origin of life, an idea stretching back to ancient Greece, known as panspermia. Academics like Francis Crick, an English scientist who co-discovered the structure of the DNA molecule alongside James Watson, argues that there is no possible way the DNA molecule could have originated on Earth. The generally accepted theory in this field, as explained above, is that we are the result of a bunch of molecules accidentally bumping into each other, creating life. However, according to Crick, we are the result of what is now known as directed panspermia. Crick and British chemist Leslie Orgill published their paper in July of 1973, hinting that we were brought here by chance or by some sort of intelligence from somewhere else in the universe. This is interesting because then you can get into a lore of creation stories that exist within ancient cultures from the world. One would be our relation to, for example, what many indigenous cultures refer to as the star people. I'm not even going to go into all of that strange skeletal remains that have been completely left out of the record. The ancient giants, for example. The takeaway now. The agenda for the maintenance of the neo-Darwinian version of the theory of evolution was nothing less than to move people away from the notion of an intelligent creator and towards a perception founded in scientific materialism. In this way, those who funded and controlled scientific activity on the planet would have tremendous power. Darwin's theory may have served humanity for a certain phase of our own evolution, but now it's holding us back. It's time for all of us to pierce more deeply into the understanding of the nature of the creation of life if we are to become creators ourselves by studying the current evidence. As a group of 500 scientists asked, how cleanly and quickly can the field get over Darwin and move on, with due allowance for every Darwinist having to study all of the evidence for himself? This is by Arjun Walia, guest writer, on Humans Are Free, by Alexander Light. Please leave your comments. Subscribe, share, and ring the bell for more new videos. Thank you for your support. If you'd like to join me on my Patreon account, you will hear content not covered by mainstream media. These riveting stories will be based on my research, and I will state my opinions and give my personal insight on diverse and controversial subjects and world events not covered by mainstream media, and not supported by YouTube guidelines. So whatever I have on my Patreon, most of those will not be on my YouTube channel. Please consider becoming a member today. More of the most significant and important videos will be on my Patreon channel. Your support helps me to continue my research and keeps this YouTube channel alive. And we depend on your support, your generous charity, because we help economically challenged families here in Athens, Greece, in Kapota, and we also help the young generation with university tuition and the community around our church. Thank you.